Hello KCET champions, how are you all? I hope all my dear friends are doing absolutely great. Well, how do you think we can deny something that you asked for? So you asked for KCET questions for so long, and now we are here to help you out. And I definitely know that you have so many examinations which are ongoing, but if at all you can find some time in this busy schedule for watching this video, for trying to solve all the questions by yourselves, it is going to be really good. And I am here, Jasmine ma'am is here to help you out with biology. Please trust me on this. And my dear children, let's go ahead. I'll be giving you some homework questions as well. Very few, but it will be really good for your practice. So question number 1, my dear children, says that identify which one of the given pair of options is correct with respect to Down syndrome and Turner syndrome. So you should be making sure that it is correct with respect to these two diseases out there. So Down syndrome symptoms, short-statured individual. Do you think they are short-statured? Yes, ma'am. So this is correct. Turner syndrome, gynecomastia in man. Gynecomastia in man means breast development in man. Turner syndrome is affecting the females, my dear children. It's not the men who are getting affected over here. So this is wrong. Round head and partially open mouth in Down syndrome. This is correct for Down syndrome, but overall masculine development. Overall masculine development, do you see it in Turner syndrome? No. Okay, you do not see overall masculine development. That is there generally in the case of Klinefelter. They have overall masculine development, but they also have certain features of females. So this is where most of the children get it wrong, but this is also not the correct option. Broad palm, physical and mental development is retarded over here in Down syndrome. Yes, ma'am, this is correct. Sterile females with rudimentary ovaries. Now Turner syndrome, I told you they are females, but these females, my dear children, since they just have one X chromosome, they will be having, it's XO condition. That means they just have one X chromosome, which is why they are going to be sterile. They cannot have babies because their ovaries are rudimentary. That means they are not properly being developed. So they are sterile females with rudimentary ovary. So here in this option we see all the things are correct. But let's see one more thing. Additional copy of an X chromosome. Down syndrome, it is an autosomal disorder. It has nothing to do with X chromosome. X chromosomes are sex chromosomes. Autosomal chromosomes are the chromosomes which have the genes that is responsible for all other features rather than the sexual features out there. So basically, additional copy of X chromosome has nothing to do for here. Absence of X chromosome is definitely a condition in Turner syndrome. Okay, they have one X chromosome being lost, which is why you have Turner syndrome. So what is the correct option? Your answer for this question is C. Now you have to look for where is C. Anyway, C is C. Sometimes they give you confusion in the form of they put B over here and write C over here. So please go ahead, look for this option, okay, not the other one. So your answer for this question would be what? C. I hope you have got it. And my dear children, one very important task. So over here I have brought 30 questions, right? You have to tell me who are those who got 30 on 30. And put it down in the comment section. Okay, put this down in the comment section that, ma'am, I got 30 on 30, I got 25 on 30, I got 10 on 30. Well, I do not wish that to happen, but yes, you have to be honest with me and you have to let me know. Okay? Chalo. Next question. A colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be. First of all, let's get stuck over here. Colorblind man. A man to be colorblind, then obviously he just has one X chromosome and that X chromosome should be affected. So usually when it's a condition like this, my dear children, when one is affected, if at all the other X chromosome is normal, it will overshadow the effect, and which is why the females who are like this, they are just carriers. They do not have color blindness. But over here in the case of male, there is no one to overshadow the effect, which is why in this condition also they will be colorblind. Now this man marries a carrier woman. That means a woman like this. A woman who is like this. So the percentage of their colorblind progeny in the next generation. Let's see, can they produce colorblind progenies? So this and this fuses, you get XXC. She is a carrier. She is what? A carrier. She has a normal gene, right? Now this fuses with this, you have XC. Here both of them are affected. There is no one to overshadow. That means this particular child has the disease. This female has the disease. Now this and this fuses, you get XY. That means it's a normal male. It's a normal child over there. So I'll just write normal male. Now this and this fuses, you get XCY. Over here again the condition like the father itself. So obviously they have to be diseased. So if at all you see, how many colorblind progenies are there? 2 out of 4. 2 out of the 4 are colorblind. You will not consider carriers. Only the people who have disease. So 2 out of 4 means half of 4 is 2. So it will be 50%. Your answer would be 50%. I hope it is also clear. Done? Dear children, if at all you are understanding, please click on the like button, share it and subscribe to the channel for more such videos. So when a single gene exhibits multiple phenotypic expression, the phenomenon is called a single gene. My dear children, we have studied one gene is there that is responsible for one character, ma'am. But over here I am telling this particular gene is not just responsible for one character but also 2 character or probably 3 character. What do you call this type of inheritance as? Polygenic inheritance. Polygenic means it has multiple genes. Poly means multiple. Genes means you know. Polygenic means multiple genes are there and these genes, my dear children, all of them are affecting just one character. For example, your skin color. Your skin color is not determined by just one gene. Multiple genes action determines your skin color. Same goes with your height. Human height is example of what? Polygenic inheritance. So with one question you know 2 things now. Polygenic inheritance, example are skin color and also height. Incomplete dominance, that has nothing to do with incomplete dominance. Incomplete dominance means, for example, I have capital R, small r in Snapdragon. It is not completely dominant over small r. So capital R is for red, small r is for white. But here, if at all there is a genotype like this, it will be pink in color. Why? Because it is not completely dominant. So this is not anyway the case for that. Now pleiotropy is there. Is pleiotropy the answer for this? Yes, it is. Pleiotropy is a condition where one particular gene, my dear children, is responsible for multiple character. This is what you see in the disease Phenylketonuria. You will be having a question related to Phenylketonuria where I will be explaining more about it. Okay, I'll be explaining more about pleiotropy out there. But as of now, one gene with multiple character is pleiotropy, not codominance. Codominance means, for example, in blood group, I have IA, IB. Both of them are equally dominant, which is why I have AB blood group. I have AB blood group. So anyway, the answer for this question would be C. It will be what? It will be C. Now let's go ahead with question number 4. How many types of gametes will be formed by a parent with genotype this? So how many type of gametes are going to be formed if a parent has a genotype capital A small a, capital B small b, capital C small c. Now this is not given in NCERT. For this you should know a formula, that is 2 raised to the power n, where n determines the number of heterozygotes. Heterozygotes means the one basically where this and this alleles are different. So capital A small a, this is a heterozygote, so 1. Capital B small b, another heterozygote. Capital C small c, another heterozygote. So how many heterozygotes do you have? You have 3 of them. So according to the formula, 2 raised to the power 3 is equals to what? 2 into 2 is equals to 4, 4 into 2 is equals to 8. Your answer would be 8. So 8 different type of gametes are going to be formed. 8 different types of gametes are going to be formed from a parent like this. Cool? Now, next question. This is related to Mendel's dihybrid experiment. So, in a dihybrid cross, dihybrid means you are considering 2 characters at once. For example, the shape of the seed and the color of the seed. So, in a dihybrid cross between a true breeding round and yellow seeded plant. So, true breeding means both the alleles are going to be same. Round and yellow. Over here, both the alleles are going to be same. That is what you mean by true breeding, round and yellow. And true breeding, wrinkled and green. Wrinkled and green. Okay. Pea plant. The ratio of segregation of round and wrinkled seed trait in F2 will be. The ratio of segregation of what you call round and wrinkled in F2 will be. What do you think is the ratio? Any idea? So my dear children, we have studied, we need not cross all of it. Rather, we have studied 9:3:3:1. Agreed? Where 9 is having the phenotype of the dominant parent. That means you're talking about round and yellow. Agreed? 1 is having the phenotype of the recessive parent. That means it has to be wrinkled and green. Now here I have 2 other things. So here it is round and yellow. Let's mix round with green now. New combination. Round and green. This is known as non-parental character. This you did not see in any parents. And here wrinkled was with green. So let's make wrinkled with yellow. Wrinkled with yellow. Now, you are being asked in F2 generation, this is for F2 generation. In F2 generation, what is the ratio of segregation of round and wrinkled? So, round is how many? 9 plus 3, 10, 11, 12. So, round is 12. Okay. What about wrinkled? 3 plus 1. Here also wrinkled, here also wrinkled. So, wrinkled is basically 3 plus 1 is equals to 4. If at all you want to take the ratio of this, my dear children, 4 into 3 is equals to 12. Ratio comes out to be what? 3:1. Ratio comes out to be 3:1. I know you need time to process, so pause the video, look out for yourself. True breeding, round and yellow. True breeding, wrinkled and green are being asked. Okay, we know the ratio for this is 9:3:3:1. We have studied that. Round yellow, dominant, nine of them are going to show the dominant character, one of them wrinkled character, and the recessive character, and two of them are going to be non-parental characters. Now you calculate the round and calculate the wrinkled. That's all. Your answer would be this. So these are the recent questions that have come. This came in 2024, my dear children. So I have started from the recent question and we will be going backwards. Now, test cross in pea plant is, what do you mean by test cross? So suppose, okay, read the question first. A cross between F2 tall plant and recessive parent, a cross between F2 dwarf plant and recessive parent, a cross between F2 tall plant with dominant parent, a cross between two F1 plants. So basically what happens is that, suppose I have capital T small t and I have capital T capital T. Basically both of them have the same phenotype, both of them appear to be tall. Now I do not know the plant is like this or the plant is like this. So what do? I have to go back. That means suppose it is F2, then I have to cross it with some F1 plant out there. I have to go back and cross it with an F1 plant or a plant which was before. Okay, so over here, the recessive parent is being chosen. Always. The recessive parent as in small t is going to be chosen, and I am going to make a cross between both of them. So over here, suppose you have small t and over here I have capital T small t. Let's see what do you get. Capital T small t is equals to capital T small t, this and this, again capital T small t, this and this small t, this and this small t. So out of 4, 2 of them are going to be tall and 2 of them are going to be dwarf. Okay, now let's see what if I cross the recessive parent. I cross the recessive parent with this capital T. Here all of them capital T small t. Here all of them will be capital T small t. Which is why, what do you find? All of them, my dear children, are tall. All of them are going to be tall. So this is what you find differently. So then you realize, okay, if it is all tall, then the plant has to be capital T. If it is half of them are tall, then it has to be in this condition. So over here, why do you choose a recessive parent? If at all you take a dominant parent, it will only show the dominant trait. Recessive parent only show the trait when they are in homozygous condition like this. So test cross in pea plant is a cross between F2 tall plant and a recessive parent. F2 tall plant and recessive parent, yes. A cross between F2 dwarf plant, do you need to cross, Baba? No. A cross between F2 tall plant with dominant parent you will not cross. A cross between two F1 plants, no. So your answer for this question would be A. And in the NCERT, they have given an example of the violet and the white flowers as well. So the one flower was appearing to be violet, but you do not know violet can be like this. Violet can be like this also. Okay, so please go through that. Now, question number 7. Test cross in pea plant is. Again the same question. I'm so sorry. I'm so sorry. Okay, question number, I think. Okay, fine. Gene mapping technology was developed by, this is related to linkage and recombination. Gene mapping. What do you mean by gene mapping? So my dear children, I am saying this gene is over here, this gene is over here. Okay, location of the gene is being identified. This is known as gene mapping. Who gave it? Correns, Sturtevant, Mendel, Schemark. It was Sturtevant, my dear children. Sturtevant basically came up with gene mapping. How? So basically his teacher, who was Thomas Hunt Morgan, okay, he was doing the experiment to prove the chromosomal theory of inheritance, and he came to a conclusion that basically genes which are far away from each other, Genes which are far away from each other, they basically show more amount of recombination. As in, suppose the condition is like this. And here there is a swap between the lower portions, okay? So this was the actual condition. Here there is a swap that is happening. So the new combination that you get is what? Capital B, small a. And over here you would be having small b and capital A. Don't you find new combinations out there? So that means recombination is possible more when the genes are far away located, when they are located far away. So with the percentage of recombination, suppose this much percentage recombination is happening, that means they are this much far. And with that information, Sturtevant, his name was Alfred Sturtevant, the student of Thomas Hunt Morgan, came up with a term that is known as gene mapping. That I can tell you the location of it, if at all you can tell me the percentage of recombination out there. Got it? I hope you got it. So it was none other than Sturtevant. So I hope my students also someday would be coming up with something. I know I might not be a good teacher like Thomas Hunt Morgan, but still. Okay. So yes. Going ahead, question number 9. Which of the following statement is correct? Change in whole set of chromosome is called aneuploidy. Do you say the change in whole set of chromosome is aneuploidy? No, my dear children. One loss of chromosome, gain of chromosome, that is known as aneuploidy. Change in the whole set of chromosome, that is polyploidy. So this is wrong. You have to tell what is correct. And whenever you're solving questions like this, underline what you're searching for. Sickle cell anemia is a quantitative problem. Is it a quantitative problem? In sickle cell anemia, what is happening? The hemoglobin which is made, the quality of it is not good. Because hemoglobin, my dear children, is made up of two things. Hemoglobin which is there is made up of alpha globin chain and beta globin chain. Over here, the beta globin chain has some mutations in it. Okay, it has some problems in it. So obviously, the combination of both won't turn out well. The hemoglobin would not be nice. The quality of it is reduced, my dear children. So here, it cannot be said that it is quantitative problem. It is actually a qualitative problem. Now, female carrier for hemophilia may transmit the disease to sons. The female carrier for hemophilia is just like color blindness. There also it is X-linked recessive disorder. So she is a carrier. Now, do you think the carrier can pass the genes to the sons? Yes. Because from mother, anyway, listen, from father, anyway, for a son to be born, you need to have Y chromosome coming. Because he is the only one who can contribute Y chromosome. From the mother, it can be either this X chromosome or this X chromosome. Suppose this comes. Then it is what? XY. So female carrier for hemophilia, color blindness may transmit the disease to the sons. Can it be wrong? No, it cannot be wrong. So this is a correct statement. It is absolutely correct statement. Now thalassemia is a qualitative problem. Is it related to quality? Thalassemia, my dear children, happens because again, there also you have alpha and beta globin chain. For coding alpha globin, you have 4 genes. Suppose one gene is mutated, alpha globin will be still produced, but not very much. The quantity is reduced. Suppose 2 of them are being gone, then again it is being produced, but the quantity is still less. Same goes with beta globin chain. There are 2 genes producing it. Suppose one is being affected, other one is still there. If both of them, the quantity of it reduces. So when the quantity of it reduces, the amount of hemoglobin also reduces, which is why it is a quantitative problem, not a qualitative problem. So this is again wrong statement. Please know where are the genes for alpha globin located, where are the genes for beta globin located. Alpha globin located on chromosome number 16. Chromosome number 16. Beta 1 is located on chromosome number 11. Chromosome number 11. Okay, so I remember it like this. Beta looks half like 6. So it should not be 16, rather it should be the opposite. Beta looks like 6, but it is not 16th chromosome, it is actually 11th chromosome. The 6 goes with A, alpha. Okay? Chalo, let's go ahead. The affected male in the pedigree chart is symbolized by affected male. So affected male is always done by a square, the colored square. If it is a plain square, that is not affected male. This is an affected male. This is what unspecified gender is there, but this gender is affected. Whatever organism, whatever human being is there is being affected. We do not know if it's a male or female. That means it's a normal female. Okay? Now, examples for non-Mendelian disorder. What are Mendelian disorders? So, my dear children, whenever there is a chromosome and in that chromosome, one gene has some mutations and because of this you have some disease. This is known as Mendelian disorder. Now, there are another category of disorder, that is chromosomal disorder. For example, you lose one chromosome, you gain one chromosome. That is why you have some disease. That comes under chromosomal disorders. Okay, something that has to do with the chromosome number, quantity and all of this structure, that is chromosomal disorder. If at all it has something to do with the genes on the chromosome, it will be what? It will be non-Mendelian, it will be Mendelian disorders. So over here you are asked example for non-Mendelian. That means you are basically asked about chromosomal disorders. You are indirectly asked about chromosomal disorders. So what are the chromosomal disorders? Thalassemia? No, it has something to do with the genes, alpha globin gene and the beta globin gene. Wrong. Hemophilia, again the gene. Again the gene. It is on the X chromosome. The gene is on the X chromosome. So this is also wrong. Cystic fibrosis. This is also what do you call autosomal disorder. That means it has to do something with the genes only. Down syndrome. Down syndrome, autosomal Mendelian disorder, okay. So Down syndrome is also an autosomal disorder, but over here the number of one autosome is extra. So that means we should be having a pair of 21st chromosome, but over here we have one more extra chromosome out there. Trisomy of 21st chromosome is happening over here. This is a chromosomal disorder. It has to do something with the number and structure of the chromosome. So Down syndrome is the example of non-Mendelian disorder. Let's go ahead with the next question. Just give me a second. Okay, so XO type of sex determination and XY type of sex determination are the examples of XO, my dear children. That means you just have one X and the other one is not there. Okay, one X and other one is not there. Well, this is usually found in insects such as grasshopper. So I'll tell you what exactly happens in them. So the females of the grasshoppers are having XX chromosome, the males are having XO chromosome. That means there is no chromosome up over here. Okay, the gamete will have no chromosomes. Suppose this and this comes together, you get XX, again a female, this and this XO, that means a male. This and this XX, again a female, this and this XO, that means again a male. So over here, males are producing two type of gametes, either X or Y. Females are not like that. So this is an example of heterogamety. Two type of gamete, heterogamety. Male heterogamety. Okay, so this is found in grasshoppers and things like that. Now talking about XY type of sex determination. Again, this is the males who have it. Females are always XX over here. In the case of human beings, we are like this. They produce either X chromosome or the Y chromosome. So again, males are producing two different type of gametes, which is why it comes under both of them. They comes under male heterogamety. So ma'am, apart from human beings, are there some other organisms showing this type of thing? Yes, Drosophila is an example for this. Drosophila also shows XY type of sex determination. Grasshopper and the insects show this type, XO type of sex determination. Now ma'am, is there some organism where female produce two type of gametes? Yes, it is there, and that is in the case of birds. In the case of birds, my dear children, the females which are there, okay, the females which are there, they produce two type of gametes. Do you know how? So males will be ZZ and the females are ZW. So either Z chromosome or W chromosome. So you can remember W for women, and that is found in birds. So females show female heterogamety only in this condition. Okay. So this is an example for male heterogamety as discussed above. Now that is a grasshopper. Chalo. Which among the following characters selected by Mendel in a pea plant is a recessive character? Inflated pod. Inflated means the pod is full. So pod inflated is the dominant and recessive is constricted. They are dried. So this is a dominant trait. So I'll make a cut. Green pod color. So whenever you go to buy peas, don't you pick the green pod color plant? Yes, right? Or do you pick yellow one? It is a green one. So green one is dominant, so this is again wrong. Whereas I'll tell you, the seed which is there, okay, we actually have green peas, but the yellow ones are the dominant ones. Anyways, white flower. There are two type of flowers, violet and white. Violet is dominant, white is recessive. So here recessive is being asked, so this should be the answer. Axillary flower. Now flower can be of two types. One flower from a place when the branch comes off, this is known as axillary flower. One which comes at the tip, this is known as terminal flower. Which one is dominant? This one is dominant. Okay, axillary flower would be the dominant one. Yes, the accelerated one will be dominant, terminal will be recessive. So white flower is the only one which is recessive, which is why that goes as the answer. Let's go ahead. And over here you have a list of all of them. Remember, seed color, seed shape 2, flower color, flower position 2, pod shape, pod color 2. Only one is there, stem height, which is single. Okay. Now match the column 1 with column 2. Column 1 is this, column 2 is this. Autosomal trisomy. That means some autosomal. How many autosomes do we have? So out of 23 pairs of chromosomes, 22 of them are autosomes. Only the 23rd one which is there, that is the sex chromosome. It can be XX, it can be XY. Suppose you have some extra autosomes out there, okay, you call it as autosomal condition. So autosomal trisomy means you have some extra autosome. 3 chromosomes are there. So as I said, in Down syndrome, 21st chromosome should be 2, but one more is there. So it is trisomy. So autosomal trisomy is shown by Down syndrome. A goes with 4. A goes with 4. I have just 2 options. Let me cancel these 2 options. Now let's go. I usually say in match the following question, go with the D option first, so that you know first and the last one. So cystic fibrosis, my dear children, is just given as a small line in your NCERT. It is not briefly explained. So cystic fibrosis, again, it is an it is a Mendelian disorder. Okay, it is a Mendelian disorder, autosomal recessive disorder. So over here, cystic fibrosis goes with Mendelian disorder. It has something to do with the genes on the chromosomes. So D goes with 2. I have the answer already with me. But still, let me see the options. Allosomal trisomy. Allosomal means sex chromosomes. Sex chromosome also have trisomy, ma'am? Yes. In the case of Klinefelter syndrome, instead of having XY chromosome, they also have one extra X chromosome. So this is known as allosomal trisomy. Allosomal trisomy is found in Klinefelter syndrome. B goes with 3. Now allosomal monosomy, that means you should be having 2 sex chromosomes, that is XX, but here in this case the female has only 1 X chromosome, other chromosome is not there. So it is monosomy. So allosomal monosomy, Turner syndrome. C goes with 1. So here your answer would be D. Okay. One more disease like this is there, myotonic dystrophy. So that is also Mendelian disorder. It is not explained, but the pedigree chart is given. It is an autosomal dominant disorder. In your NCERT, no dominant disorders are being mentioned except this myotonic dystrophy, okay, or the muscular dystrophy, sorry. Going ahead, we have the next question. The genotype of a husband and a wife are, husband genotype is IAIB and the wife's genotype is IAIO. Okay. Among the blood types of their children, how many different genotypes and phenotypes are possible? How many different genotypes and phenotypes are possible? Let's see. So this fuses with this, I get IA. This is the genotype. The phenotype is what? A blood group. Both of them are AA only. This and this fuse it, I get IAIO. A is dominant over O, which is why it will be A blood group itself. This goes with this, I get IAIB. Both of them are co-dominant, so it will be AB blood group. This and this goes, I get IBIO. B is also dominant over O, which is why it is B blood group. So if at all you talk about genotype, all of them are unique. IAIA, IAIO, IAIB and IBIO. All of them are unique. So how many genotypes? 4 genotypes. 4 genotypes. What about phenotypes? Both of them are A, so I will consider it as 1. A, B, B. 3 are there. So phenotypes, if you see, 3 are there. So 4 genotypes and 3 phenotypes, I have the answer as B. I have the answer as B. Done? Done. Next, match column 1 with column 2 and find the correct answer. Aneuploidy, monoploidy, polyploidy, diploidy. Aneuploidy means, I told you, loss or gain of a chromosome is known as aneuploidy. Okay? So A goes with 2. A goes with 2. How many options do I have? I just have 1 and that gives me the answer. But let's see the other options. Monoploidy. Monoploidy means what? Monoploidy is not there in your NCERT. Polyploidy is there. Polyploidy means increase in the whole set of chromosome. This is usually seen in the case of plants. So C goes with 1. Now monoploidy means, my dear children, you just have a single set of chromosome. You should have 2, but you just have a single set of chromosome. So that is monoploidy. Diploidy means you have 2 sets of chromosome. So that is this, diploid 2, right? So that is why your answer would be C. Next question. A pure breeding pea plant with round and yellow seeds was crossed with a pea plant having wrinkled and green seeds. So pure breeding means again round and yellow like this, and wrinkled and green seeds means it will be like this. Okay? So on selfing of F1 hybrid, 64 progenies were obtained in the F2 generation. So in the F2 generation, you got 64 progenies. Okay? Find out the number of F2 progenies showing non-parental character. That means something that was not found in the parents. Ma'am, this is super easy. We studied 9:3:3:1. I told you 9 of them are going to show the dominant parent's character. So it is going to be round and yellow. Agreed? This is going to be showing the recessive parent. So wrinkled and green. Now both of them are going to show non-parental character. So round was with yellow, but here round will be with green. And here wrinkled was with green, but here wrinkled will be with yellow. It will be with yellow. Now these two are the non-parental character. So how many? 3 plus 3 is equals to 6. 3 plus 3 is equals to 6. Out of what? 9 plus 3 plus 3 plus 1 is equals to 16. So 6 out of 16 are what? Non-parental. But you have to find it out of what? 64 progenies. So what do? You do into 64. So 16 into 4 is equals to 64. Right? 6 into 4 is equals to what? 24. So out of 64 progenies, 24 of them are going to show the non-parental character. Got it? Easy. So out of 16, 6 into 64. That's all. If you're asked about parental character, then 9 plus 1, 10. 10 by 16 into 64. Got it? Yes. So this is not pleiotropy, this is regarding the dihybrid cross. Okay, next. In an organism, mutation in a single gene exhibits multiple phenotypic expression. Identify the underlying genetic mechanism. I told you, a single gene, if at all, it is guaranteeing multiple features or the multiple character. This is an example of what? Pleiotropy. We studied about it directly, pleiotropy, and I told you Phenylketonuria is an example for this. Do I have a picture? No, I do not have a picture. That means you have one more question. So pleiotropy is an answer for this question. One gene, multiple character, pleiotropy. Polygyny, multiple genes for one character. Now in the following symbols used in human pedigree analysis, identify the symbol that denotes consanguineous mating. What do you mean by that? Mating without consent? No. Mating between relatives. So you should be having one square, one circle, and in between that you should be having two lines. So one line means it's normal. Two lines means it is mating between relatives. So two means more close. You can remember like that. So your answer for this question becomes this, B. Okay? Okay. 20th question. In Morgan's experiment with Drosophila, when yellow-bodied, white-eyed female was crossed with brown-bodied, red-eyed male and their F1 progeny were intercrossed, what was the percentage of recombinants in the F2? What? Yellow-bodied, white-eyed male. Yellow-bodied, white-eyed male were crossed with brown-bodied, red-eyed male. How much percentage of recombination was found? So it is less amount of recombination, that is 1.3. I'll tell you why. So over here, always remember, yellow-bodied gene and the white eye color gene, they are very close to each other. They are very close to each other. And what do you have? You have brown body and red. Brown body and red eye color. These two genes are also close. So suppose I have a swap. So both of them will be swapping together. They are closely linked genes. They are closely linked genes, my dear children. So which is why even if the swap, my dear children, both of them are going to go together. So the percentage of new combination which will be there will be very less. So out of this, very less is what? 1.3. Okay, 1.3. And I'll tell you, 37.3% recombination, where do you see? I'll tell you. So yellow-bodied, white eye, and then suppose over here you have one more gene which is coding for the wing. Okay, the wing. For the wing gene, it is located very far. So if at all you are asked, what will the percentage of recombination if these two characters are given, then it is going to be 37.2. Why? Because they are far away from each other. So if there is a swap, there is new non-parental characters that you will be able to see because they are far away from each other. They are loosely linked. Over here, these two features, always remember yellow-bodied, white eye. They are tightly linked, which is why less recombination is there. That is 1.3% recombination. Maximum is 50. Here 37.2 is shown when wing and eye color is given. You understand it is more. 21st question. From the chromosomal complements given below, identify the one who shows female heterogamety, where females are going to produce two types of gametes. This is a condition found in insects. This is a condition found in human beings and Drosophila. This is the XXY means Klinefelter syndrome and XX means normal female. So over here, this condition, my dear children, is where you find female heterogamety. Here females, my dear children, are going to produce Z and W type of things. Where do you see it? I told you, you see it in birds. Okay? Next. Both male and female have normal vision, though their fathers were colorblind. Father colorblind, write it somewhere. Father has to be colorblind, that means the condition is like this. And the mothers did not have any gene for colorblindness. They didn't have any genes for colorblindness, means both their chromosomes are very, very normal. They are not even carriers. The probability of their daughter becoming colorblind. Now, if you want daughter to be colorblind, my dear children, you should be getting one X chromosome from this mother. Okay, that is affected, and one X chromosome from this father also, that is affected. But here mother's chromosomes are not affected at all. Then do you think you can get a female who is colorblind? You will always get a female who can be a carrier. From here this, from here this. So she can be a carrier, but she is not going to be colorblind. So the probability of their daughter becoming colorblind is equals to what? 0. Man to be colorblind, he has to be like this. Mother is not having any gene for colorblindness, so she should be normal as well. Got it? Okay. Now choose the possible genotypes responsible for the lightest skin color in human beings. I told you skin color is determined by multiple genes, which is why it is an example of polygeny. Same goes with your human height. So multiple genes means, ma'am, over here multiple, suppose like this. So I am saying capital A is here, capital B is over here, capital C is over here. That means the genotype is capital A, capital B, capital C. This means that person who has this has the darkest skin tone. Suppose it is not capital A, rather you have small a over here. Then it turns out to be like this. This person will have a lighter skin tone than the other person. So ma'am, you are saying the ones that will be having the recessive, all of them the recessive, they will be having the lightest skin tone? Yes. And that is the question. Choose the possible genotypes responsible for the lightest skin color in human beings. That has to be C. Small a, small b, small b, small c. Okay? Okay. Now, the type of sex determination in honeybees. That we didn't study. In honeybees, so basically, my dear children, in honeybees, the diploid ones, the ones which has 2n number of chromosome. Suppose honeybees usually have forgot their chromosome number. 32. Yes, they have 32 number of chromosomes. That is diploid condition. And half of it is what? 16. Suppose if someone has 32 number of chromosome, this will be a female. And if it has half the number of chromosome, this will be a male. This is known as haplo-diploid sex determination. Haploid male, diploid female. That is usually seen in the case of honeybees. Now if at all you're worried, how does it work? Listen to me carefully. So in the case of females, they are diploid. If they have to form gametes, they will be undergoing meiosis. Half the number of chromosomes will be there, 16 over here, right? Now if it is a male, my dear children, they are going to not undergo anything because they are already haploid. They only have 60 number of chromosomes. So they just undergo mitosis to increase the number of cells. So over here 60 number of chromosomes are there in the gamete. Suppose this egg carrying 60 number of chromosomes and this male sperm carrying 60 number of chromosomes comes together. 16 plus 16, 32, it will be developed into a female. But what about the other one which is not undergoing fertilization? This egg containing 60 number of chromosome, if it is not undergoing fertilization, it undergoes a process that is known as parthenogenesis. Parthenogenesis, where without fertilization it will be turning into a male. This is known as haplo-diploid sex determination, something which is very unique. And this is found in the little honeybee that you see. So haplo-diploid sex determination in honeybees, do not forget it. Okay? Okay. Now in a dihybrid cross, when F1 plants like this are self-hybridized, the ratio of segregation of yellow and green in F2 is. So F1 plants like this are self-hybridized. That means if you got F1 plants like this, so their parents might have been like this. Agreed? They were like this, then only you got something like this. It is there in your NCERT itself. Now what is the segregation of yellow and green? Same thing as we did. Same thing as we did. Round and yellow, wrinkled and green, wrinkled and yellow, round and green. How many yellow are there? 9 plus 3 yellow. So 12 yellow. How many green are there? 3 plus 1. 4 green. So it will be what? 3:1. If at all you are asked yellow and green ratio, so yellow is 3 and this is 1. 3:1 will be your answer. Same question repeated again after so many years. Okay, we saw that. So it was talking about round and wrinkle trait then. So the same principle applies over here. Okay, so don't get confused. Now, question number 26. Example for autosomal hyperaneuploidy. Aneuploidy means loss or gain. And autosomal. There's only one example. Down syndrome is where you get an extra autosomal thing, a gain of a chromosome. So Down syndrome would be your answer. 21st chromosome basically comes under autosomes. 23rd chromosome are basically what? Your sex chromosomes. So this is your answer for the question. So over here you see a tiny little extra chromosome out there. So all of them are in pair like this. Okay, 21st is one extra. This is a 23rd chromosome. Now, from the following pedigree chart of a family, you tell what is this? What is this pedigree chart denoting? Autosomal trait, dominant trait, recessive trait or sex-linked trait, what is it? First of all, very easy to find dominant and recessive. Suppose it is there is skipping of generation. Okay, there is skipping of generation. For example, this generation does not have it, but as you see, this generation is affected. There is skipping of generation, right? There is skipping of generation. In that particular case, my dear children, it will be a recessive disorder. It has to be a recessive disorder. Suppose in every generation at least one parent is affected, then you call it as dominant disorder. But here in this parent is not affected, here it is. Okay, the progeny is being affected. So yes, you find skipping of generation. Now here the parents, both of them are affected, my dear children, parent and progeny, but if at all you see, they are not affected, which is why you see skipping of generation. Now, it is autosomal or is it allosomal? That means it is sex-linked or it is autosomal. How do we define that? So over here, okay, over here the male and the female, they are mostly affected equally in this generation. Okay, usually in X-linked disorders, the males are generally more affected. But that is also, reason also if we leave. We usually see crisscross pattern. That means suppose a female is being affected over here, then a male has to be affected, then a female has to be affected. That is a crisscross pattern. But over here you only see a female being affected. Okay, so crisscross pattern, if at all you see it, it has to be sex-linked or allosomal. If you do not see it, then it will be what? Autosomal disorder. And it is autosomal recessive over here, which is why this is the answer. B. So skipping of generation, recessive. Crisscross, then it will be what? Sex-linked. But over here, you are not seeing any crisscrosses and you are seeing both male and female are almost equally affected in one generation. So it has to be autosomal recessive disorder. Next, the gene for hemophilia is located on X chromosome. Hence, it is normally impossible for. Okay, it is normally impossible. The gene for hemophilia is located on X chromosome, which is why it is normally impossible for hemophilic father to pass the gene to his daughter. So this is hemophilic father. He can pass the gene to his daughter because this is the only gene that he can pass to the daughter. So this is wrong. It's not impossible. Carrier mother to pass the gene to her daughter. Carrier mother is there. She can also pass this to her daughter. Anyway, one X chromosome has to go, so this is also wrong. Carrier mother to pass the gene to her son. Again, mother will give X chromosome, so it can be any other X chromosome. This is also wrong. Hemophilic father to pass the gene to the son. Hemophilic father. Can he pass the gene to the son? No, it has to be the Y chromosome that has to be affected if he has to pass. So this will be wrong. So hemophilic father to pass the gene to his son is practically impossible. Okay? Fine, that is why I said crisscross pattern. It's an X-linked disorder. Fine, so males, females, and then males, females, or females, males, and then males, like that it goes. Now, 29th question. The gene disorder phenylketonuria is an example for. The gene disorder phenylketonuria is an example for, and this is your homework question. I'll explain what is phenylketonuria. I'll tell about everything, but you have to tell me what is the answer for this question. 29th homework, put it down in the comment section. I'll tell. But here I showed you the answer. Then how can it be a homework question? So then my plan is flop, but anyway, listen to me. So there is an enzyme, okay, there is a gene in the chromosome that is responsible for the production of the enzyme phenylalanine hydroxylase. Now when it is not being produced because of the mutation in this particular gene, what happens? Phenylalanine, which is an amino acid, is not being able to convert to tyrosine, which is why it gets accumulated in a body in the form of phenylpyruvic acid. And this is very toxic for us. It can lead to mental retardation. So this is one feature. This is one character being affected. Next thing, if tyrosine is not being produced, it cannot help in the production of melanin, which is responsible for our skin color, hair color, lot of things. Now if melanin is not being produced, we get hypopigmentation. Hypopigmentation means it looks very whitish and your hair turns grey in color. Okay, so this is the next character being affected. So one gene being affected, two things are being affected. One gene, when it is being affected, two characters are also being affected. So which is why this is an example of what? Pleiotropy. Okay, it is an example for pleiotropy. Now, the last question for the day. Now you might be wondering, one question was repeated, ma'am. So in the next session, I'll bring that one more question, don't worry. So find the incorrect statement among the following. In sex-linked recessive traits, the gene is transmitted from unaffected carrier female to the male progeny. What? So you have to find incorrect, okay? Incorrect. In sex-linked recessive trait, the gene is transmitted from unaffected carrier. Unaffected carrier means it is this to some of the male progeny. Is it? Yes, it can be. It can be. So this is correct. You have to find what is incorrect. Accumulation of phenylpyruvic acid in brain results in mental retardation. We just discussed about it. Yes, it does. Individuals affected by Down syndrome will have congenital heart defect and are more intelligent. So yes, they have congenital heart defect out there, that is genetic, but they are not more intelligent. This is where you lose your marks. You just read this and you see, oh my God, this is anyway wrong. Turner syndrome is caused due to absence of one X chromosome. This is correct, no? There is only one X chromosome, there is absence of another X chromosome. That is XO condition. So your only answer becomes C. Your only answer over here becomes C. Okay? So they are not more intelligent, rather they have mental retardation as well. So, how many of you got 29 on 29? If that is the condition, please let me know. And my dear children, I hope you had understood whatever I taught you. And if at all you did, definitely do not forget to hit on the subscribe button so that you get lots of sessions like this. And let me know how frequency should be increased or it's okay. Okay, you want more videos coming after KCET or not? Let me know in the comment section. See you really, really soon. Take care. Bye-bye.